friends, and today's welcome lecture. This lecture is on protoplast isolation and regeneration, and the course is Plant Biotechnology. Code is RBT602 Biotechnology. So let's start this topic, and for ease, I'm dividing this topic into four different subtopics. So we will be studying this topic under these heads. Like first, we're going to talk about protoplast. What is protoplast, and what are the important features of protoplast? Second, we're going to talk about protoplast method of isolation of protoplast. Third, we're going to talk about the culturing of protoplast. And fourth, it will be the development and regeneration of protoplast. So let's start this topic. I'm trying to keep it short as possible so that you can find it easy for your revision as well. So let's start this topic. First, the question is, what is protoplast? So you should know that protoplast is a cell without cell wall. So in plant, if you remove the cell wall, then what remains is called the protoplast, or alternatively, you can also call it a naked plant cell. So by definition, it becomes cellular content excluding the cell wall, or you can simply say the cell divided of minus cell wall is called the protoplast, or you can also call it a naked plant cell. Now, such type of cell which has no cell wall over it, what is the utility of that, and where we can find its application? So it becomes the important features or the properties of protoplast, so that it can easily uptake the genes, or we can use it for fusion purpose, so that we can fuse the two protoplasts to make a hybrid cell. So that we're going to learn in protoplast fusion. Again, it is an important feature of protoplast, or the property of protoplast, that they can be fused. Okay, so that helps in reading that helps them in taking the gene. Second is that the same protoplast, the hybrid protoplast, we can easily regenerate into fertile plants. And if it is a fusion product or the hybrid product, then also we can regenerate plants from it. And thus we can find a way to regenerate a hybrid plant from the this fusion product. Now, these protoplasts also have some important features and the potential, like they can regenerate a cell wall, and it is an important feature once we expect some regeneration or differentiation division from the single cell like the protoplast. So first they regenerate a cell wall, then they undergo dedifferentiation to form a callus. So they keep on dividing themselves mitotically, and then they proliferate and make a callus first, and further they undergo differentiation to make shoot and root to make a complete plantlet, and sometimes the same callus became embryonic to give rise to somatic embryos, and from those somatic embryos we can produce a complete plantlet. That will be the hybrid plantlet if we have undergone the fusion of the protoplast, and if it is simply a protoplast cell, then we will going to get a clonal plant propagation through this method. So protoplast has enormous application in plant biotechnology, and one of the major application of the protoplast in plant science is to produce a hybrid plant that we call somatic hybrid or the hybrid sourcing. That will take in another lecture, and in today's lecture, after this protoplast introduction, let's talk about protoplast isolation. The second task comes here is the method to isolate the protoplast. So what method we use to isolate protoplast? We can divide them into two categories. The first one is the enzymatic method, and the second one is the mechanical method. Now, in enzymatic method, as its name indicates, so we use enzymes, certain enzymes to isolate the protoplast. Now, by now you understand the meaning of protoplast. It is the cell without cell wall. So if you want a cell without a cell wall, that means we need to remove a cell wall. So for removing that cell wall, we have two methods. And if we apply these two methods in which we actually remove the cell wall, what we'll get is the protoplast. So all the two methods are actually the method of removing the cell wall, so that we can easily isolate the protoplast from the cell mix. So or the material or the explant. So first method is enzymatic method. Now, if it can be easily guessed that in this particular method, we must be using some enzymes to isolate the protoplast. That means we are using those enzymes which actually remove the cell wall, or in alternative words, we can say that these are those enzymes which degrades the cell wall. Okay, so let's understand how this method actually works. So in this method, we actually use certain enzymes, and these enzymes which actually helps in dissolving the cell membrane, cell wall, and leaving the protoplast intact. So in this particular method, we use two ways to apply the enzymes or certain enzyme which actually degrades the cell wall. So these are the two ways. Like the first one is the sequential hydrolysis, and the second is the mixed enzymatic hydrolysis. And in sequential method, we will first be checking sequential method. In sequential isolation, we actually add enzymes in a sequential way. That means first we use some enzymes, and second time we will apply certain enzymes so that the method actually needs some protoplast there. So firstly, the cells are separated by using the maceration enzymes. So remember the name of these maceration enzymes, the pectin hydrolyzing enzymes such as mesozyme or pectolyase. Because you know that cell wall is made up of pectin, so we use the enzyme which hydrolyzes this pectin, and they are in cell wall, and the name is mesozyme and pectolyase. And once the cells are separated, that means the cells are loosened now. They washed in CPW solution free of enzyme but containing plasmolyticum by gently centrifuging at 100 G. So it is very low RPM, and we keep this RPM just to collect the cells there. And the pellet is retained and suspended in the second enzyme like cellulase or hemicellulases. Again, you know that cell wall remains on the protoplasm. Cell itself can be washed properly with the or can be hydrolyzed with this two enzymes, cellulase and hemicellulase. So we used to hydrolyze the remaining cell wall component, and once the protoplasts are released, they are washed with CPW to remove the debris. Okay, so it is very simple step in which we apply the enzymes. First category of enzyme is mesozyme, and the second category of enzyme which we use to remove the cell membrane, cell remains or wall remains onto the cell. So in mesozyme or mesozyme enzymes are mesozyme and pectolyase, and the second category of enzymes are the cellulase or hemicellulases. So these two enzymes we add one after one so that the method becomes a sequential isolation method. Second is the mixed enzymatic approach. Now we have same enzymes, but here now we are applying them in a mixed way. So in this, plant tissues are plasmolized in the presence of a mixture of pectinases and cellulases. So we are using the pectinase enzyme, which helps in dissolving the pectin layer to loosen the cells, and cellulase enzyme, which actually dissolves the cell wall component, that means the cellulase and hemicellulases. So thus including simultaneous separation of the cells and degradation of the was to release the protoplast directly in a single step. So the only difference in these two steps or the method are that the way we use the enzyme. In sequential isolation, we use the enzymes first, the pectin isolating enzyme, or second we use the cellulase or hemicellulase enzyme. And in mixed enzymatic approach, we use both the enzymes simultaneously to get the protoplast isolated. Now here you can see in this picture that whatever the protoplast we get, okay. So let's detail this that we are having this. In this, we have two plants, like suppose plant A and plant B. So we would be isolating the protoplast from both the plants, and the purpose behind isolating the protoplast is to fuse the characters of these two plants with the help of protoplast fusion method. So first we'll be isolating the protoplast, and we have taken the leaf as an explant. So both the plant from both the plants we are taking leaf as an explant material, and leaves we actually subject to some isolation protoplast isolation method. So what we get is the isolated protoplast. Then we apply some method to fuse these protoplasts to get some fusion product, and these fusion products are further differentiated, further cultured onto suitable medium to first produce the callus, and from callus we can find the regeneration and differentiation there, so that we can find a complete plant that produce. So in this way we can find the hybrid plant produced from a hybrid plant produced from this protoplast isolation and fusion method. So it is the sole purpose of isolating the protoplast, and in a nutshell we have seen that what we will be doing after isolating the protoplast. Now, we should know that we are isolating protoplast using certain enzymes. And if you are using enzymes, then conditions are very important because enzymes actually works in certain conditions. So what conditions are required for enzyme activity? We should know that enzymes are pH and temperature dependent. Thus, for enzymatic release of protoplast, an enzyme showing the activity at pH range 4.7 to 6 and temperature range of 25 to 30 degrees centigrade is used. So, ideal temperature and the pH conditions are here. And during duration of enzyme pre-treatment and condition of light presence required for incubation may also be determined. So, it is again matter of research to see that whether the pre-incubation period and other things like light and dark period, if it is necessary for enzymatic treatment to isolate the protoplast. Enzymatic mixtures used should essentially consist of cellulose, hemicellulase, and pectinase enzymes, which facilitates the degradation of cellulose, hemicelluloses, and pectin respectively. So, pardon it is cellulase is here. So, we use cellulase enzyme, hemicellulase, and pectinase enzyme, which respectively dissolves the degrades the cellulose, hemicellulose, and pectin, hemicellulose and pectin respectively. So that these are the major components of the cell wall. Concentration of sugar alcohol used as osmoticum or manicum must be empirically defined. So this sugar solution is used in order to maintain the osmolarity so that the isolated protoplast won't get burst or shrink there. So in order to maintain the osmolarity of the solution, we use the sugar solution there uh, um, in this method of enzymatic isolation. Okay, so it is very simple method in which you can use the enzymes easily remove the remove the cell wall components and isolate the protoplast. Now the second method is the mechanical method. Now, as its name indicates, in this mechanical method, cell wall were capped in suitable plasmaloticum, for example, CPW solution containing 3% weight by volume, mannitol. Once the plasmolysis is complete, while remaining in the osmoticum, the leaf lamina would be cut with a sharp edged knife. So we'll be using some knife to actually chop down the explant material to remove the to in order to isolate the protoplast. And plasmarate cells were cut through the cell wall. So whatever the cutting is there, if it actually happens so that the wall is actually being cut and no damage is actually happening to protoplast, so with that cut, the protoplast will leak out from the cell. And we are actually there will be the least intact protoplast there, while some of the protoplast may also be damaged inside many cells. So it is just a method in which we get no more success, no major percentage of success because the failures are very high. So whatever the protoplasts are intact there, we can collect them. So this method seems to have some disadvantages, and disadvantages are like it yield in lower protoplast there. So that means the yield lower protoplast yield is there. It is a labor-intensive method. Protoplast obtained has very low viability, and method is applicable only to vacuolated cell. So not onto all plants we can apply this method to isolate the protoplast using the mechanical method, though it is easy and cheapest method, but again the method yield is very low. So we can find enzymatic method a superior one over the other methods. So in these methods, we have discussed two methods, enzymatic approach. In enzymatic approach, we talked about two methods. First is the enzymatic approach, in which we add the two methods, sequential and the mixed enzymatic approach. And second one is the mechanical method. Now, the second is the factors affecting the yield and viability of protoplast. Again, we should know that if we are trying to isolate the protoplast using any one of the methods, then what are the factors that affect the yield and viability of protoplast? Now, again, as we're dealing with the plant, so source of material does matter, and the source of material says that leaves were the most convenient source of the plant protoplast. So it has been researched that leaf, if you are using as an explant material for isolation of protoplast, they will give the best result compared to other explant material. And the parent plant age and the conditions in which it is growing have profound effect on the yield of protoplast. So you should also remember that the conditions like the plant age and other things, the environment in which we are growing it, also affects the yield of protoplast. Cultured cells can be used as a source material. Now, it is very important again if you have already have some plant in cultures and we are experimenting protoplast isolation, we can experiment on them. So it is better to use the cultured material because the protoplast yield will be better compared to other filled material. Okay, yield of protoplast depends upon the growth rate and growth phase of the cells. Again, you should remember that not all the cells are in the same growth rate or growth stage severe phase, so the protoplast yield also is affected there. Generally, embryonic suspension cultures are used to obtain totipotent protoplast. Okay, so if we generally embryonic suspension cultures, okay, if we have embryonic suspension cultures, so we can have some totipotent protoplast also, which can actually give rise to the complete plantlet using the somatic embryogenesis approach. Second is the pre-enzymatic treatment. As it was mentioned in the enzyme treatment method, then pre-enzyme method also has effects on protoplast yield. To facilitate the preparation of enzyme solution into the intercellular species of the leaf. So in this method, we actually treat the cells or the material, external material in such a way so that enzymes can easily penetrate into the uh, spaces between the cells. So peel the lower epidermis and coat the strip pieces on the leaf on the enzyme solution in a manner that peeled surface is in contact with the solution. So this is the way we actually pre-treat the material using before applying the enzymes there. Cutting the leaf or tissue into smaller strips. Okay, this will also help in attaining more contact of your material with the enzymes. Agitation of incubation mixture during the enzyme pretreatment improves protoplast yield from the cultured cells. Okay, so these three ways are there. If we use some lower epidermal cells and then just float them onto enzyme solution, so that will help us in making the good contact of enzyme to these cells, so that we can find the good separation of the protoplast there. Then cutting of the leaf or tissue into smaller strips can also help, and agitation can also help in treating the material with the enzyme solution in order to get the best yield. Then enzyme treatment itself has effects on protoplast isolation like cellulase or pectinase enzyme, and the activity of enzyme is pH dependent, which we have already seen. So cellulase and pectinase enzyme is required to digest the cellulosic cell wall, and the pectinase is mainly degrades the middle lamina. Some of the tissue may require other enzymes like hemicellulases and other mesozymes or petrolases. And since we know that enzyme is pH dependent, so we should also be careful about this. pH of the enzyme solution is adjusted somewhere between the 4.7 to 6. So these are the factors that affects protoplast yield and viability there. Then choice of plasmolyticum. Now we know that why do we need plasmolyticum and what is it? So sugar solution, mannitol or sorbitol we use to maintain the osmolarity of the solution, and sometimes the salt mix of protoplast was ingredient. So Cockings per day and Peverday and White. So after these three scientists, the name of solution is CPW solution. And the sugar solution on mannitol or sorbitol that we use. So mannitol is usually preferred since it is not metabolized by the plant cells. And once the protoplast divides and regenerates the wall, no more osmoticum is required there. So you should remember all these points. And it should be removed gradually from the medium, otherwise cell division stops. And it slowly removes the osmoticum from the medium. The protoplast can be isolated in high osmoticum mixture consisting of both mannitol and sucrose. The sucrose will be metabolized by the dividing protoplast and thus will reduce the osmolarity of the medium. Normally, mannitol is used as concentration range of 11 to 13%. So why do we use mannitol and why it is preferred over the sucrose or other uh, solutions? So it is pretty clear here. Now, CPW solution is the composition of the CPW solution. You can just remember it, and if we are actually going through the practical experimentation, then we have to make all this solution with this different composition. It has potassium dihydrogen phosphate, potassium nitrate, calcium chloride, magnesium sulfate, potassium iodide, copper sulfate in all these different composition, and it is for 1 liter. The amount is mentioned here. The pH is 5.8, and this particular solution we actually use as a plasmolyticum solution. Okay. We have this picture to explain about the method we use in isolation of protoplast. So we take leaf as an explant material. We can also take sometimes the callus or the in vitro cultures as an explant material to isolate the protoplast. Whatever the material we take, we actually chop it into smaller pieces as we have seen in the pre-treatment method that actually enhance the yield of the protoplast. So we chop the material into smaller pieces, then incubate it with the enzyme solution, and the osmoticum is there so that the protoplast cells won't get damaged. After isolation won't get damaged. So there is some incubation period, 16 to 18 hours. Such is incubation period is there. So we find the protoplast settled at the bottom of the dish, and the enzyme solution is removed. So we remove the enzyme solution then the protoplast are transferred in a washing solution. Again, it is a CPW solution. Then we centrifuge it so that all the protoplasts will settle at the bottom of the tube, and the remaining supernatant and the tube top will going to be discarded. Now we again resuspend them in a washing solution, sucrose washing solution, and we centrifuge it. So cell debris will settle at the bottom of the tube, and the protoplast obtained after centrifugation will float at the top of the supernatant. Now we actually take out this protoplast uh, and from this tube, and we resuspend them in culture medium. Now once we resuspend them in culture medium, now it is, remember that now it is culture medium in which we are going to culture these protoplasts actually. So we can go for counting of these protoplasts. We resuspend we suspend them in more culture medium to give a perfect plating density. And afterwards, we use some tissue culture techniques to culture these isolated protoplasts. Now, up to this point, we have seen that how the protoplast is isolated and further subjected to culturing. We have not talked about any culturing method or culturing technique, and we have also not talked about the protoplast fusion. So up to this point, this protocol says how do we isolate protoplast, how do we go for uh, counting these protoplast, and how do we actually subject them to culture. Okay, so after the protoplast isolation, the next task comes where we need to culture these protoplasts. Now, for protoplast culture, there are methods available, like the liquid method is there, and these are those methods in tissue culture which we use for the culturing of the isolated or the single cell. So, protoplast is none other than a single cell, and we need to apply those tissue culture techniques we use for the single cell culture. So, it is a liquid culture method or embedded in agar or Agros method, feeder layer, and the co-culturing method. Okay, but just one by one, we're going to see the details of all this method. Liquid method. So in this method, we prefer easier dilution and transfer of the. Now, the important feature of this particular method is that it provides easier dilution and transfer of the thing. The osmotic pressure of the liquid medium can be effectively reduced after a few days of culture. The cell density can be reduced, or cell in a spatial interest can be isolated easily in liquid medium. So we can easily isolate the cells of interest from this liquid culture. The protoplast suspension is plated as a thin layer in battery plates, incubated at static culture in flask, or distributed in 50 to 100 microliter drops in battery plates, and is stored in humidified chambers. So it is one of the simplest methods we can use to culture the isolated protoplast cells. Then embedded in agar and agarose. So we can again see some of the important features of this method. Like this method of agar culture keeps the protoplast in fixed position. So since we are embedding them in agar or agarose, so they are in a fixed position, thus prevent it from forming the clumps. Immobilized protoplast give rise to clones, which can further be transferred onto other mediums. So whatever the embedded cells are there, they keeps on dividing there, and once they divide, then they make clone of them. So we get some cultures there which you can easily isolate from the embedded agar plate. In practice, the protoplast suspended in more than 40 degrees centigrade agarose medium, 1 to 2% weight by volume agarose are dispensed 4 ml into small 3.5 to 5 centimeter diameter plates and allowed to solidify. So these are the technical details of the method. The agarose layer is then cut into 4 equal sized blocks and transferred to larger dishes containing the liquid medium of otherwise the same composition. So we first prepare some plates in which we actually add this protoplast solution there, and then we just keep on multiplying them on try transferring them into the suitable agar plate. So in this way we can find the cells cultured there. Then is the feeder layer culture. In order to culture protoplast at lower density, okay, remember that if you want to culture them, if we have a very low density of protoplast initially in the isolation stage, then this method is preferred. So feeder layer technique is adopted. A feeder layer of X-ray irradiated non-dividing but mitotically active protoplasts after washing are created in soft agar medium. Now here you can also note down that we are also using these protoplasts in order to produce certain mutants. And if we are using some method of creating mutation in these cells, so we can automatically find the lower protoplast density there, and such a treated material has to be cultured if then the feeder layer culture is the best option. And the non-irradiated protoplast of low density are created over this feeder layer. The protoplast of the same species or different species can be used as a feeder layer. So we used to use some of the cells which are radiated ones on the cells which are non-irradiated one. Non-irradiated are the protoplasts of the desired type, and irradiated are the one that we are using as a feeder layer there. Okay, so we can easily find this protoplast cultured over the feeder layer. Co-culturing. Now, co-culturing means that this method involves the co-culture of protoplast from the two different species to promote their growth or that of the hybrid cell. So, metabolically active and dividing protoplast of two types, slow and fast growing, are cultured together. Fast growing protoplasts provide other species with diffusible chemicals and growth factors which helps in cell wall formation and cell division. Okay, so we are using the two types of the protoplast here. The co-culture method is generally used when calli arising from the two types of protoplast can be morphogenically distinguished. For example, protoplasts isolated from albino plants and green plants are easily distinguishable based on color, where albino protoplasts will develop more green, non-green colonies. So it is just a technical detail to show you that how this method will work. So so far we have seen the four methods of culturing the protoplast. Liquid method is there, embedded in agar or exhaust method is there, feeder layer is there, where if the initial feeding density is low, then we can use this method, and the co-culturing is there. So here it is shown in pictures that how these four methods actually can draw a diagram of these four methods. Protoplast culture in liquid medium. So here in this, like this protoplast we actually culture and on in a liquid medium. Protoplast culture on a semi-solid medium. Okay, protoplast culture with some feeder layer there, which is irradiated cells and cut down as a feeder layer, and over it we are using the non-irradiated cells, which we actually want to grow further. And then we are using some co-culturing method in which the fast-growing protoplast is there, slow-growing protoplast is there, which are actually providing some essential nutrient and the growth factors to these growing cells. Okay. Second is the after technique is the culture medium. So what culture medium is there? Of course, mostly it is Morashican school medium and the P5 Gambrox medium that we use for culturing the protoplast. Second is the ammonium salt, which has been found detrimental to protoplast survival for many species, and medium have been devised that either have a reduced concentration of ammonium or lack of it. So by experiment, scientists have already proven that ammonium salt is detrimental to the growth of these protoplasts. So we need to use a medium where the no ammonium salt is there, or it is having some alternative sources there. Concentration of zinc is reduced while the concentration of calcium is increased as it enhances the membrane stability. Third one is osmolarity. Osmolarity is maintained by addition of sorbitol, as we have seen previously, mannitol, glucose, and sucrose, and mannitol being widely used as osmoticum, as it is not used by the dividing cells, thus maintains osmolarity of the medium. Now can easily, you can please underline this thing that why we use mannitol as one of the most commonly used osmoticum, because it is not used by the cells. Glucose is preferred carbon source as sucrose do not satisfy the protoplast culture. Again, it is actually the outcome of the research that scientists have found that glucose is actually preferred as carbon source. Again, the growth regulators used, we use certain oxygen cytokinins and sometimes both in combination in order to proliferate the dividing cells and the protoplast cells so that we can find a callus produced from the protoplast cells. And later on, we actually vary the concentration of these PGR in order to find some organogenic or somatic antigenic response. So again, in nutshell, you can say that high concentration of oxygens because initially we want to get we are expecting some callus from the cells, callus formation. So we will be using some oxygens like NAN240, which is the main calcium hormone, and we use lower concentration of cytokinin initially. Then the environmental conditions. In environmental condition, we should know that high light density or intensity inhibits the growth of protoplast. So initially, protoplast is grown in dim light for a few days and then transferred to light of about 2000 to 5000 lux. However, better results are obtained when culture is in darkness. So we know that initially the light is required or it is essential for the protoplast growth, but gradually we see that we need to remove the this thing, light intensity. The splitting density, as it is mentioned previously, that splitting density has its role there because sometimes splitting density is high, then we can use some one or other method of protoplast culturing. So protoplasts are cultured at a density of 1 into 10 to the power 4 to 1 into 10 to the power 5 protoplasts per ml of the medium. And at high density, cell colonies arising from the individual protoplast tends to grow into each other, resulting in chimeras. So if you don't know about chimeras, then please wait for the next lecture. So high plating density will result in chimera tissue formation. If the protoplast population is genetically heterogeneous, then it is a major concern here because you don't want the protoplast to show some weird characters there because of some random fusion there. Cloning of individual cells, which is undesirable in somatic hybridization and mutagenesis study, can be achieved if protoplasts or the cells derived from them can be cultured at low density. So regarding the protein density, we know the ideal protein density. We know what will happen if the protein density is high. It will result in pseudo combinations there, or hybrids there, or chimera formation will be there. Then what if the protein density is low? We know the benefits and the drawbacks of having the lower high trading density there. So remember this point also regarding the protoplast culturing. Now the protoplast development and regeneration. Up to this point, we have seen that what are protoplasts. Let me just revise it once again for you. So we have seen what the protoplast is all about. We have talked about the protoplast isolation. We have talked about the method. Methods are two: enzymatic method and the mechanical method. Now, beside the method, we have also discussed about the factors that affect the yield and viability of protoplast. It is the source of material, pre-enzyme treatment, and enzymatic treatment. Then second thing we have seen is the choice of plasminoticum, how the sugar alcohol or menthol soluble effects, and salt mixture of CPW solution effect. Then we have seen this picture to show you that how the protocol isolation actually works. Then protoplast culturing, again we have talked about the method of techniques of protoplast culturing. We have four methods here: liquid culture, embedded in agar of agarose culture, feeder layer, and the co-culturing. So with this diagram, it is clear that how this four method actually works. Then we have talked about the culture medium and the plating density. Now last thing we are going to discuss here is the protoplast development and regeneration. So further development and regeneration is again a typical micropropagation protocol where we have some material with us and we just want to produce a plant from it. Now here the material is a cell. It can be a hybrid cell or a hybrid protoplast cells. So whatever the material is there, so we actually culture them in a suitable medium using one or other method that we have seen previously. So we will find that protoplasts regenerate a cell wall within a few days, like 2 to 4 days of culturing. And protoplasts lose their characteristic spherical shape, which has been taken as an indication of the new wall generation. So wall generation is usually there, and the characteristics spherical shape of the protoplast will get lost. There is a direct relationship between wall formation and cell division. Protoplasts which are not able to regenerate a proper calli will fail to undergo normal mitosis. And the other important point here is to note here is the once the cell wall formation is complete, undergo division, resulting in increase of size of the cells after an interval of 3 weeks. So after the 3 weeks, we're going to find these cells perfectly dividing there, and then the colonies will appear, and these colonies will further going to be transferred onto suitable medium, which is free of any osmoticum, and will find the callus induction there. Okay. From callus, as you know, as we have studied earlier, that from the callus you can directly go towards organogenic response, or you can directly find the plant produced from the scalars using the embryonic response if your material is having that embryonic potential there. So that's all about this topic. Hope you interesting, and I'll come up with remaining topics from the subject of plant biotechnology. And you can comment whatever the social media you're using to watch this particular video. Okay, thanks for watching.